cannabis to save the world is maybe not such a far-fetched statement after all, right?
Yeah, not right now.
Well, talking about saving the world, right now, I think that's the appropriate time to bring in our next guest.
Yeah, we're super excited to be joined from Canada by Igor and Olga. You guys have been at the cutting edge of research into COVID and cannabis. Just where did that story start and get going?
Good morning, afternoon, or good night. I'm not sure where you are watching from, right?
Just hello.
Yeah, so, before starting on COVID, we worked on cannabis for the past 5 years, right? So researching various medicinal properties, breeding different hybrids, testing anti-inflammatory, anti-cancer properties, aging, skin conditions like psoriasis, atopic dermatitis. Recently started diabetes model, tissue fibrosis models, stuff like that. And when COVID started, we already had a large data accumulated that we initially just reanalyzed what we had in our hands, and with the notion that we're hoping that cannabis and cannabinoids would influence the expression of the ACE2 receptor, so basically a gateway receptor for the virus. And we indeed found a number of our hybrids substantially decreasing the expression of receptor in various tissues, as well as we found several cultivars that significantly, substantially decrease the expression of all pro-inflammatory molecules, so hopefully it can prevent the cytokine storm development in COVID patients.
So I guess, just to try and translate that a little bit, I guess it sounds like your research is proving positive so far.
Well, in the laboratory, yes. So we have had a lot of very exciting and interesting results in the laboratory setting, using cell line models, using tissue models, which are very special tissues that are specifically grown in the dish to model exactly how human tissues are. So, excuse me, in those, actually, we do have quite some positive and encouraging results. But of course, to have full confidence, these need to be further extended into clinical studies, right? Before anything can be rolled out and recommended. Because right now, this is based in the laboratory. But again, I think it has a lot of future and a lot of potential in it.
Sounds amazing. So you're developing tissue samples, and then are you putting COVID into those tissues, infecting it with COVID, and then how are you adding then cannabis or cannabinoids into that to test it?
Well, sorry, no, we do not give them COVID because one very important thing, SARS-CoV-2 research is research of biosafety level 3. And actually, before the pandemic hit, there were very few, if any, biosafety level 3 labs that were still commissioned at the universities. Again, because this is for working with certain types of human pathogens. So for that being said now, so what we do, we induce inflammation because as any pathogen, what COVID does, it induces massive amount of inflammation. And there are other, other ways to induce inflammation in the tissues, and this is what we used to. You can use it by UV exposures, by hydrogen peroxide, certain types of concentrations, or by simply treating them with tumor necrosis factor alpha in the cocktail of certain pro-inflammatory molecules, and then we treat them with extracts or extra components to see whether or not we are affecting that inflammation. That being said, we also started a collaboration with actually a virology lab, and keeping fingers crossed, so they would be teaming up with us to see whether there are any direct effects on the virus.
Wow, nice. And how many different ratios of cannabis extract did you test out? I had a read through the study, and I see lots of different ratios of cannabinoids between THC and CBD there. How many did you try, and what did you find was more effective?
Right. So I would say we tried a couple dozens from those already that have been pre-analyzed for their anti-inflammatory properties. And we found that the ratio doesn't mean much, right? So it does not reflect the activity of an extract, but rather the terpene profile does, right? So as soon as it's a high CBD extract, it has to have a specific terpene profile to be valuable for COVID research. And different terpene profiles would be good for different things. So one, for example, terpene profile would be good to decrease the ACE2 and TRPV receptor expression. So assuming this would be preventative, right? So, and another one, a different profile would be to decrease inflammation in cytokine storm. Completely different cultivars. They would not be useful in both cases, but in one specific only.
Wow. Are there any standout terpenes that we should be looking for when we're shopping for our cannabis then?
I don't think these strains exist. Excuse me, anywhere. They're mostly proprietary strains from our lab, but when we try to work with companies in US, for example, to do clinical study, we quickly realized it would be almost impossible to move a product from Canada to US. It's just very time consuming and lots of logical problems. So instead of that, we have reconstituted top 4 or 5 terpenes in best varieties, and scientists and doctors in US found the variety that closely approximates this formulation and just added terpenes so that it mimics our variety as much as possible. And right now we have a study going on to develop a mouthwash, like an OTC, over-the-counter, for prevention of COVID and for reduction of symptoms and complications and stuff like that. So.
Go on, Kelly.
So, like, oregano has been shown to have terpenes in it that help with coughs. Has there been any correlation between the sort of the terpene profile of the cultivars you were using there on this study and things like oregano, for example?
I don't believe they have the same terpene profiles, but I'm not familiar with terpenes of oregano, frankly, so it's hard to say. But a lot of like bergamot, for example, right? So limonene, yeah. So it's a very common that exists in many places. But there are some specific. Well, even pinene is not specific to cannabis, but something like myrcene, caryophyllene. So they are more specific, right? And it's not only specific set of terpenes, but their concentration, their ratio seem to be very important. We're now doing this reconstitution experiments where we try to see the effect of single terpenes and then their combination in addition with CBD. And there is a very substantial difference. So terpenes at least double the effect of CBD on the ACE2 and in the expression of pro-inflammatory cytokines.
Have you been looking at any of the flavonoid content as well within the plants? everybody started to wake up to the terpene idea and knowing that this has this effect. But was any of the flavonoids looked at and the amino acids? Forgive me, was it a full plant extract that you used within the studies, or was it isolates that you added terpenes back into?
Right. We haven't looked at flavonoids at all, so mainly cannabinoids and terpenes. And yes, it was a full flower ethanol extract, right? So basically a solvent-based extract that brings all the molecules into it, right? But when we did reconstitution experiment, then we used single ingredients to basically reconstitute what we found in the cultivars.
It'd be interesting to see how this turns out. I'm very excited to see how it you know, what the actual compounds are.
Yeah, I just wanted to ask what stage your research is at now, Igor. Obviously, it needs to get peer reviewed, or is that the next stage for you and your paper?
Both have been peer reviewed.
Okay.
And both have been accepted for publication. And the only thing is, usually all the journals, they first announce themselves. So I think it would be kind of wrong to steal their thunder to say, kind of the embargo that they usually all have. So, but what I, excuse me, because we've already seen the proofs. And so for the first one and the other one, we just signed all the paperwork for, permission for publication. So I really hope it should be coming up. The first one probably this upcoming week, and then the next one will follow, because as I said, it was a long road. We had to do additional experiments. There were some very interesting suggestions that were given by the reviewers. So we had to kind of, go above and beyond and try to, do additional experiments, bring in additional tissue models to kind of, substantiate. And yeah, we did it all. It took quite some time, but.
And what I just add to that, because I've heard Kelly's comment on the low impact factors of journals that a lot of this research is published. So these went to the journal with impact factor higher than 5. So it's a very, very strong journal. When it's published, there will be no doubt. So.
I was just wondering, do you imagine you could have got so far with this research, or if you'd have been, anywhere but Canada? Has legalization helped your research? Because before you guys joined, we were talking about how in this country still doctors just don't have the access to the research and the understanding.
You're right. So I imagine this could only be done in Israel or Canada. I don't think anywhere else you could do that. So.
And just out of interest, how did your journey with cannabis start? Was it something you'd always taken an interest in, professionally, or?
Right. So, we both have medical degrees, right? And in addition, well, it's a long story, but I switched to plants, so became a plant biotech person, and my interest picked in medicinal plants. So this combination of medical profession and plants basically brought me to medical plants. And I used to work with coffee since 2009. I had a dealer license from 2010 to work on restricted substances. And when cannabis came to being, so in 2015, it was kind of no-brainer because we already had a cage, a vault, and everything. So yeah, it was simple. Yeah, simple. Yeah, it took about 12 months, but I remember to get a license to work on poppy, it took almost 3 years.
Right. And I guess, are you seeing, as researchers then, as medical people, what do you think about the plant? Is it are you seeing benefits for all these kind of ailments that people assume?
Well, I've heard you guys talking today, and you're absolutely right. So this is a plant we have co-evolved with, right? So we have been, and the plant has been around as long as we have been around. And the there is a substantial difference between the endocannabinoid systems in different organisms. So it's more elaborate in animals, more sophisticated in mammals, but human is most versatile, right? So, and I believe that's a reflection of the past probably 100,000 years of continuous consumption and selection of the expression receptors in the entire endocannabinoid system. So it's a basic homeostasis system in the organism. So it regulates and balances your response to stress primarily. So if it's a physiological stage, it doesn't do much. As soon as something is brought up by stress, it will bring it down. If it goes down, it brings it up. So it basically balances your responses and makes it most efficient. And therefore, as you correctly said before, I think Kelly did, said it doesn't have a unique response, so that you have to bring these molecules up in order to have the response. You just add cannabinoids, especially if it's a whole spectrum, and the organism will decide, because there are a multitude of receptors through which they work. The organism will decide how correctly to use these molecules. We can store them in fat, so it means that we don't have to metabolize and use everything. So maybe, if we do gel cap, we consume, let's say 10% becomes bioavailable, another 5% will be stored in your fats, right? So, and if you do it regularly, it will be stored regularly. So the system balances and takes only the amount that needs to balance itself and to function the best. That doesn't, this is not a right expression of a characterization of the addiction drug, right? So
yeah, wow, yeah, I've never heard that. That's blown my mind. That's yeah, really exciting.
So we did at uni was in your hair. It's stored in your hair for up to 3 years. So the storing of them, you can detect that there.
My dad always thought that was why cannabis was a dangerous thing, because nothing that stays in your system for so long can be a good thing. That's just my dad's logic, right? He's coming around. He's coming around now, as I know a lot of parents, and we're having those conversations. But it's just funny. It's just like weird science. Well, it's not science, but it's just like weird applied logic that doesn't really make sense.
Yeah, and metabolism of every person is very unique. So if you take how quickly the drugs are metabolized, we have about 20-fold difference in the spectrum. So one can, say metabolize in 10 minutes, and another person would do it in 200 minutes, wow. Right? So, and that means that, the rate of metabolism often reflects how high the expression of receptor is, how much molecule you need, that what is the longer term response to this molecule, whatnot. So every organism, every individual balance it, right? So if your receptors are higher, and your metabolism rate would change, and so on and so forth. So that makes, cannabis consumption very individual, right? So, and probably heard that titration is the best. There is no better way how to test, but go small and slow, right? So, and then increase until you know that it works.
Yeah, start low, go slow.
Yeah. Most favorite thing said in this industry: start low, go slow.
Absolutely, and that's even in Canada here. That's what the official government line is. Like start low, go slow, and improve from there. And it does make sense. So, what's the kind of end goal with this research? You mentioned like maybe a kind of CBD or cannabinoid infused mouthwash, which might coat some of the areas with the ACE2 receptor that you mentioned. What do you think is the scope of that? Because right now, of course, everywhere you look, they're talking about vaccines. You've got a couple of these big pharmaceutical companies seemingly race. It seems a bit rushed to bring out a vaccine. Do you see this as an adjunct or potentially like replacement for the need of something like that?
And I believe the answer lies in will of governments and pharma industries to pick it up, because if the pharma against it and the government against it, it will have very low and limited impact, right? So that's how it's been for a long time. A lot of companies that we work with, they're on a smaller scale, right? So we haven't been approached by Pfizer or whatever, right? So those are those companies who work with us. They're all small scale, US and Canada, but we have signed agreements on the patches, on inhalers, on nasal spray, mouthwash, maybe I forget something. So 4 different modes of delivery for as a prevention of COVID or treatment. I believe it could be used for both, right? So right now, there is really no effective other drug for COVID, unfortunately. And if you look at the effect of CBD and terpenes, it rivals dexamethasone, right? So as Israeli researchers showed that dexamethasone was inferior to CBD alone and inferior to CBD with terpenes, and even inferior to terpenes alone in reduction of pro-inflammatory cytokines. This is mind-blowing. This is the drug that they push on us for decades and decades. And this is the life-saving, supposed to be drug that should be given to COVID patient. And here we like walk in with bunch of terpenes. So basically, take a lemon peel, pineapple, I don't know, squeeze it, and here's the drug better. Like that just shows you what pharma does and how they make something shitty, right? So, and they sell us high compared to what's available in nature. And that's the only reason why it's not allowed, because it's too simple, it's too cheap, right? So it can be used by anyone. If we release all these formulations, any person can go into the store, pick up some herbs, some fresh fruits and vegetables, squeeze, make juices and whatnot. I may be exaggerating, it's a bit more complex than that, but the idea is that it would be fairly straightforward. And look at the price of CBD right now. In US, they offer a kilogram of CBD isolate for $500 US. $500 US. The prices went down 20 times in 2 years. Mm-hmm. And so if you take the price of $500 per kilogram, and if you need 100 milligram dose for one single CBD pill, it becomes affordable, right? So we are basically right now at the stage where technology and terpenes, we were buying, I think for $1,000, we bought in the lab, let me see, 100 milliliters, 12 bottles of 100 mil terpenes. So over a liter for $1,000. So 1 liter of terpenes would probably last as medicine for years for me, right? So that means just theorizing, I can buy for $500 1 kilogram of isolate, buy those terpenes, and make myself medicine for me and my family that will last me for years of protection from cold and whatnot. And obviously, no pharma wants this, right? So.
Yeah. Well, that's incredible. And terpenes avoids it's not cannabis, right? So it's not regulated in any country that I'm aware of. Just terpenes. Otherwise, you'd have to regulate lemons and most like fruit and vegetables and plants.
That's actually true. And I know the original question would you ask, if I remember, whether or not it will become an adjunct therapy. I think that would be probably the first step to get it through as an adjunct therapy, because proving superiority, again, this is a strong uphill battle, which will just require huge amount of regulatory hurdles and huge investment to overcome those. Albeit, if you can say this is an adjunct therapy, this is probably the easier way of trying to, go through, get the trials and start introducing it. But you're absolutely right, because again, the availability of CBD is not becoming, it's not an issue anymore, right? It's not outrageously expensive. So it is there. It's just the availability of the will to do it. Because usually, as we say, if there is a will, there is a way, right? And I think thus far, we still need that will to materialize in more serious ways, that more trials can be done, and then doctors actually, because otherwise doctors can't recommend. If we don't have a bona fide trial, doctors can say, oh yeah, use this because there was something that was done and it helped 3 laboratory mice. I'm joking, right? For example, right? They need more evidence than that. So, and so we need that willingness to conduct that research and those clinical trials.
Yeah, well, good luck, guys. It's so exciting to hear what's happening at the forefront of cannabis medicine, especially with the COVID and the pandemic and whatnot. It seemed, when I first heard about some of the research, it seemed like another reason just to legalize or give people access to it. Like, why are you having to jump through these hoops, which I'm not sure some of the vaccines are having to do, but
Yeah, I think CBD has to be regulated as much as vitamin C, vitamin D means it should not be, right? So very clear that it's safe. So there's plenty of information what it does and what it does not, right? So just the recommendation to people, as I said, so the titrate, even CBD has to be titrated, right? So, but yeah, it should be on the shelf of everyone. So.
Nice.
Amazing. Well,
do you know what your next? Have you got any research plan going forward? Are you still going to be focusing on cannabis for a while, or?
Yeah. So we continue working on cannabis. As I said, as soon as we started reconstitution experiments, we found some amazing results. So as I said, there are very specific ratios of terpenes, and the story is coming. So hopefully, within 3-4 months, we'll finish that. That has tremendous effect. So our purpose is to find the terpene profile that would be as close as possible to full flower extracts in terms of efficiency. So even if we can reconstitute efficiency by 70% just using terpenes alone, so suddenly we're not scheduled drug anymore. So that means any country can start preparing it, right? Yes, of course, it will be produced from cannabis primarily, but when it's shipped as mix of terpenes. So that means the countries who produce it, right? So whether it's US or Canada or Israel, can ship it to any country in the world or give them formulations, right? So it should have a big impact.
And just finally, then, for anyone, there's a few people commented saying, "Hey, how can we follow your work?" So how can we follow your work as this research unfolds?
Yeah, unfortunately, we're not really part of any media or whatnot, so we don't have Twitter or whatever account. So
and we're happy to share it with our audience as well, for sure. So let us know any new milestones that come up, and we'll absolutely share that out, because we're all pretty fascinated in this research. It's great to see, and I think we need more researchers like you guys as well. So thank you very much for the work
for joining us.
Great job.
Yeah, thank you for doing it. It's really great way of popularizing also cannabis and cannabis research. It's really,
it's exactly how it started in Canada. People talked, people became, probably shouldn't be saying it online, people become less obedient in following what the government asked them to do. and the government was forced to change. So do what you do.
Thank you very much. It's been lovely joining us. See you soon.